Organizing this workshop on emerging technologies has been really cool, and I'm excited for the other talks coming. So I'm a physician scientist, psychiatrist at Stanford University, working in the Pascal lab, and I'm going to talk to you today about my work on human brain organoids. Specifically, I've been working on developing the technology further to allow them to basically generate much greater diversity of neural populations than we have been before, and also investigate the limitations in maturation that we're seeing in vitro. So human brain organoids are emerging technology. They've been around for maybe 5-6 years. It's worthwhile asking the question of why do we need a human cellular model of the brain? And of course, there are some obvious differences between most experimental neuroscience model systems and the human brain that are clearly evident, but there are many other factors as well. So the human brain isn't just a larger rodent brain. It's different on cellular, genetic, molecular, connectivity, behavioral levels. And it's really important to note that a human neuron develops at the pace of a human, as a human does, and rat neurons in vitro develop at the pace of rat neurons. So there are intrinsic clocks to these different species that are evident when you work with stem cells. And this is all due to the 100 million years of evolution that separate rodents and humans. And you can just imagine the amount of genetic accumulation that happens between these species. There are many examples. One clear example is of a gene phenotype difference between rodents and humans that's pretty significant is the LIS1 gene that when is disrupted in humans results in really severe neurodevelopmental disorder called lissencephaly, where you have a smooth brain. The rodent brain is already lissencephalic. And so when you introduce this gene into the rodent brain, there's relatively little structural defects in those animals. And so it's just one example of, you can study genetics and do a lot of things in rodents. And I have myself too before. I have nothing against rodent models. They're really important, have contributed a lot to basic understanding of neuroscience. But as we want to get further with human neural replacement or human neurobiology, we need to start developing a human neural system. The other reason to develop this technology is not just in experimental models of understanding the brain, but also in the future for developing therapies for patients with neurodegeneration, in aging contexts, and also other neuropsychiatric disorders. So we know that neurons get lost with age, maybe globally, in Alzheimer's disease in specific regions of the brain, or in other conditions like Parkinson's disease, you have loss of really specific neural subpopulations. Furthermore, in conditions like epilepsy or autism, it's been hypothesized and that there's an imbalance of neurons that are generated in certain areas of the brain that results in these clinical phenotypes. And so in all these contexts, you can imagine a future world where we can replace neurons in specific regions of the brain, or even replace entire regions of the brain that maybe get lost in a stroke. This is obviously decades out, but right now there are clinical trials ongoing to replace specific populations. For example, Blue Rock Therapeutics is developing stem cell-derived dopaminergic neurons for replacement in Parkinson's disease. And there are also Neurona Therapeutics is developing inhibitory neuron transplantation for intractable epilepsy, so putting in these inhibitory neurons to silence networks in these patients. So stepping back, just what are we doing here with these stem cells and organoids? What is all this stuff? And so maybe many of you are familiar with reprogramming, where you can take skin cells from patients or really anybody, convert them into stem cells, and these are induced pluripotent stem cells. And from there, you can convert them into a variety of tissue types. And how do we do this? How do we know how to do this? And this is really by replicating what happens in vivo. So what happens in vivo is that we have a series of what we call morphogen gradients. So these are molecules like sonic hedgehog or WNTs or VMPs. They're secreted from organizing centers called organizers. And cells integrate signals of these different molecules into transcriptional programs, which then dictate what cell type they're going to become. And by understanding how this process goes in vivo, we can replicate this process in a dish and then generate these populations. But brain cellular diversity is really immense, and there's only so many molecules that are being used. And right now, we can only generate a certain number of cell populations. A lot of work has been done on cortical neurons or motor neurons or dopaminergic neurons, but there are many other populations you might want to replace. And so what I want to do is take a screening approach to this question of how does a limited set of morphogens generate immense amount of diversity in human cells using an organoid screening platform. And really, this is due to the identity of the molecules, how they're used in combination, the timing at which they're applied to cells, the duration of their action, sometimes also the sequence of which molecules signaling pathways get used first, and also their concentration. So I designed an organoid screen. So we take stem cells, we dissociate them and create these little balls around 10,000 cells each, almost exactly the same number of cells in each spheroid. For 6 days, we use dual SMAD inhibition, which has them differentiate into the neural lineage, so they make sure they're neural cell types. And then for 15 days, applying these different molecules in different combinations, and then afterwards letting them culture for another 50 days, all in vitro, to allow them to develop to the stage of generating a significant number of neurons. And this process is really long. We have to do this for 70 days because these cells develop at the same rate as they would in utero in human development. And at that point, we do a number of phenotypic outputs of, we do tissue clearing, immunohistochemistry, qPCRs, but really importantly, with the advent of single cell atlases of the human developing brain, we did a multiplex single cell sequencing readout for our screen, and then use these atlases to map onto them, onto the developing human brain, exactly which populations we generated. So first thing we did was we compared the single cell gene expression from a recently published fetal brain atlas for over 1 million cells and our own single cell screen from these organoids. And on the bottom left, you can see the integration of the two populations. So in gray is the atlas and black is our cells. We generate oligodendrocytes, radioglia, and lots of neurons. We covered roughly what we estimate is that 65% of human brain regions just within this screen alone. And these range from forebrain to midbrain, hindbrain populations. And on the far right, you can see the cluster overlap of our organoid screen clusters on the top. And on the left side, the clusters of single cells from the atlas. And so we have really good mapping onto which populations we're generating in our screen to which ones are present in vivo. So as I mentioned, we generated really a massive diversity of cell populations. And so on the left, I've color-coded all of the cells by their cluster identity. And again, we've generated both neuronal and non-neuronal lineages, including the choroid plexus, progenitor populations such as the cortical hem and floor plate, support cells like astroglia and oligodendrocytes, as well as tons of different neural populations in the cerebellum, thalamus, GABAergic, really a diversity of GABAergic neurons, and lots of cortical neurons as well. And so really we're just struck by the amount of diversity we were generating by modulating these different combinations. Because we did multiplex single cell, we could actually trace back for each of those cells which molecules they were exposed to. And then we color-coded them by that morphogen condition identity on the right. And what you can just see by looking at is that the colors here are also clustering separately. And that's because if you apply certain molecules, for example, KIR, which is a Wnt activator in BMP, you generate lots of choroid plexus and thalamic progenitors as well. And for oligodendrocytes, which are really blue on that side, most of those cells came from conditions exposed to retinoic acid or sonic hedgehog agonists. And so we want to really integrate all this information because this, I think one of the powers of doing this kind of multiplex single cell work is that you can start to take a more unbiased approach to analyzing this kind of complex data and looking at all kinds of correlations that you would not really be able to extrapolate by doing a single hypothesis-driven targeted approach. And what we really want to start building is an atlas of human cellular differentiation into different lineages, such that if you picked a neural population you want to generate, we could build enough of an atlas here that you could just figure out which molecules you need to apply in which combinations. In our screen, we had roughly like 40-ish conditions, but in the future, we really imagine this should be scaled up by 10 to 100 times. And even since starting this experiment many years ago to now, we could already really do the same amount of with the same amount of cost, probably scale it by already tenfold. And what this kind of atlas shows is on the top is all the cell populations that we generated, and on the left side is all the different conditions and the molecules they're exposed to. And we can start to see emerging principles, such as if we want to generate mice to positive forebrain neurons, we saw that all those conditions had something similar. They had received sonic hedgehog activation after day 11. And then further down, if we want to generate cortical hem and Calretzius neurons, we actually saw those conditions had Wnt or BMP activation, but just before day 13, meaning some of these temporal switches just come out of doing this kind of really multiplexed work. So we did a lot of other characterization of these organoids. These are tissue clearing of specific organoids. So obviously some grew very large. Those exposed to growth factors got quite large, of the largest ones that we generated. And also, because we can do tissue clearing and staining, we can also look at the structural components on the inside. And what you see here is the ventricular-like zones that are being formed within these organoids, which are these proliferative centers. And we also wanted to just really be rigorous about this and not just trust the single-cell data blindly. What we also did was took every single condition and stained them for the oligodendrocyte marker olig-2 and compared the immunohistochemistry staining percentage of oligodendrocytes on the x-axis there with the percentage of oligodendrocytes by our single-cell data. And we saw a really good correlation there. And so because we have not just single-cell data, but also this morphological and immunohistochemical data, we can start doing additional cross-correlations between these morphological and cytoarchitectural features and the single-cell identity. And one of the interesting things we found was that just because an organoid is large, it's been assumed that, that it's really related to the number of ventricular-like zones, these little clusters that are forming. But actually we found that these structural features are more related to a brain regional identity than actually its size. So for example, cortical neurons are really highly correlated with conditions that also have these ventricular-like zones, like in that FGF8 condition. But large organoids can also be generated by other molecules like FGF2, which do not have that kind of structure to them. And so in a way, we're trying to also look at the structural principles of how these things are developing in vitro as well. So just a couple of examples of specific conditions that came out of the screen was that we generated a new protocol to generate cerebellar populations by the specific addition of FGF2, but not FGF4 or FGF8, and we have validated all of this by immunohistochemistry as well. And then, as I mentioned before, we found a protocol to generate this interesting population of Calretzius neurons, which really migrate far distances around the cortical surface to regulate neural migration across the cortex. And we really saw there's a really specific timing switch by which by activation of the Wnt pathway with this molecule KIR, if you apply it before this time point, roughly around day 13 or 14, they will generate Calretzius neurons, but if you just apply it a day or two afterwards, they will not generate any at all. Meaning they're really fine switches in human development that these molecules are able to direct cell identity. And then we went further to try to characterize more of their function using what we call assembloids. And this is where we take organoids from different brain regions and fuse them together to look at migration or integration of neurons from these different populations. And so on the top, complicated side, but basically we labeled these Calretzius neurons with viral labeling of HSIN YFP, and then fused them with a cortical organoid, which is where they usually migrate. And we can quantify the amount of cells that are migrating. We see they're really highly migratory, which is what we would expect based upon their cellular identity. And then we also use spatial transcriptomics as well to characterize migration of certain cell populations where we don't have good immunohistochemical markers, such as recently described TAC3 positive population of interneurons. And we can fuse them to striatal organoids because this is actually the brain region where they preferentially migrate to. Okay, quiz time, it's on you guys. What do you guys think is the most beautiful neuron in the brain? There is a right answer, so. Anyone have a? Purkinje. Yes, good. Okay, so obviously this is correct because it sources all of human knowledge to decide which is the most beautiful neuron, and it's the Purkinje cell. It's located in the cerebellum, has beautiful dendritic branches. And so the Calretzius, Purkinje neuron, is on the front cover of Ramona Cajal's, just a recent book that came out there. And now we have, I told you I generated Purkinje neurons, and we know that they are by all these transcription factor markers. But after 8 months in culture, we're waiting and waiting and waiting for this beautiful, our beautiful front cover of our journal, and it just didn't happen. And this was really striking because we have to ask the question of, do we know what these cells are becoming? What's going on? Like, are they really becoming the cell population we think? And so this spurred our thoughts to really push the boundaries of what we're doing with organoids. And obviously with this in vitro setting, in a dish, we're feeding as much as we can. The physiology we're trying to regulate, but really, honestly, a lot of the media we use are the same media components that have been used for like 20, 30 years. We don't know exactly, there are too many components for us to engineer. And it's not going to replicate the in vivo environment. So we want to understand is there something intrinsic to stem cell-derived neurons that limits their maturation, or is it really the context in their environment? So we transplanted these cerebellar organoids into the neonatal immune-deficient rat, and we tracked their growth with serial MRIs, and then captured them with vibrotome section to get at the structure of these organoids transplanted into the rat brain. And on the right side, you can see the amount of vascularization that happens after transplantation. That these are really well vascularized. They're going to get all the nutrients that the rat brain usually gets. And what we found is that, really strikingly, these neurons do start to develop much more than we ever saw in vitro. They form also many interesting structures, like these Purkinje neurons on the left form this, they like to cluster together. We also find they're really highly migratory, which we were very surprised by. And they also form natural structures where their dendrites are projecting one side and their axons are projecting another. And it's not exactly the most beautiful, regular thing you would see in the normal cortex, cerebellar cortex, but it is striking that it's almost attempting to form a structure that, we're not otherwise directing in any kind of way. And on the right side, you can see just some imaging of these Purkinje neurons in green, that mainly showing that their orientation is, so their cell bodies are on one side and their dendrites are projecting to the other side, which is what you'd usually see in the cortex. We did lots of tracings, manual tracings of these neurons, both in vitro, comparing the same exact batch of organoids that were transplanted or kept in vitro. And we could see really striking differences again in their structure, over this transplantation period of roughly 130 days post-transplantation. And what we find is that they're not becoming adult Purkinje neurons, and we wouldn't expect them to be, because I mentioned before that these neurons generated from stem cells develop at the rate of human neurodevelopment. And so we actually see that after roughly 30 or so weeks of transplantation, they resemble what has been documented as the roughly 30-week Purkinje neuron in the human brain. And so we're continuing that maturation pace that is really severely limited in vitro. So just stepping back, what I've told you is that we did a morphogen screen using single-cell RNA sequencing. We've generated really a massive amount of neural diversity than we have before. We've generated these really regionalized brain organoids and also investigated the influences of what's really limiting human maturation in vitro and trying to break that limitation in vivo. And it's worth just thinking about this, this hypothetical future where we can generate specific brain regions and transplant them into each of you if you would ever need them. And thinking about, this, I think the most interesting thing I take away from this is that this temporal thing of, even if we transplanted these organoids into your brain, they wouldn't age match your current brain. You would have baby neurons. And how long would it take for them to reach even adolescent age? And what that means for neural replacement in the future of, can you become young again? So anyway, with that, I just want to thank everybody. There's a lot of people who contributed to this work, and so thank you.
Thank you very much. So time for 2 questions. Back in the back in orange, and here then if we have time.
Fantastic talk. You mentioned the temporal aspect, which basically Laurie Studer published an interesting ways to accelerate neural differentiation. I'm sure you're familiar with these H2 in particular. But I don't think it's tried in 3D. Have you tried any of these tricks? And I'm actually
Sorry. Need to come a bit forward.
I can repeat the question. So yeah, so he was mentioning that Lawrence Studer has been working on advanced kind of methods to force maturation to happen faster. A few groups have been studying the even basic mechanisms of how that's possible, that a species-specific stem cell, you can't generally force it to change its rate of development. I think the 2D versus 3D, they're roughly the same. I think these things will work similarly. I don't think this question is by any means solved. I think it's really going to be a much larger question that I think touches on so many areas of biology that I don't even have a clear hypothesis of how this temporal aspect of development is contained within a cell. I think it's really going to be a challenging, cognitive, for us, just theoretical challenge of understanding that, and then translating that into an actual maybe pharmaceutical or genetic therapy to advance it. I think we just don't know, and I think the work that's being done is really important, but has by no means solved the question.
Well, one of the most interesting is ARDD2 inhibitors that tells you that it's probably some of this is epigenetic, right?
Yeah, a lot of things are epigenetic, so, but we don't know where and what and how. Yeah.
Yeah, so
Yes.
thank you for a great talk.
Thank
So what's your sort of general take on partial brain replacement? And I'm thinking specifically of John Abair's work out of New York. And how does your work sort of relate to that? And what's your take on the eventual feasibility of partial brain replacement?
It's just not happening anytime soon. There's so many clinical challenges here. I mean, we do this in experimental setting, but of course there's challenges of how do you restrain their growth, potentially create cancers. It's just there's so many potential. You just ask yourself the question, would you want to do this yourself? And, I think there's so much that needs to be done to understand. This is just the biggest challenge with cell therapies in general, is that it's not a molecule that can be regulated and exactly what you're getting with a cell. You'll never exactly know, what's in those. And so I think it's just a massive challenge, and it'll take a very long time to have anyone feel comfortable with doing that.
Perfect. Thank you, Neal.
Thank you.